Hello, and welcome to today's LinkedIn Live event, Unlocked, Keys to the Undruggable Target Space, hosted by Bayer. Thank you for joining us, wherever you are in the world. Over the next 40 minutes, we will venture into the depths of the so-called undruggable target space and explore with two experts what exciting opportunities lay within that space. If you follow Bayer's accounts here on LinkedIn, you will have seen that over this week, we have spent quite some time talking about how science could open the door to a tremendous number of opportunities in the undruggable space. Today, we have the chance to talk to two experts who are at the forefront of these efforts to really unlock all these opportunities. My name is Nuria Aiguabella Font. I will be your moderator for today. And joining me are Michael Brands, Senior Vice President and Head of Drug Discovery Sciences at Bayer Pharmaceuticals, and Todd Kinsella, Head of Biology at Vividion Therapeutics. Michael, Todd, thank you very much for joining us today. For those of you who are listening to this panel, you are very encouraged to put your questions on the chat. Let us know what you would like to hear about, how you would like to shape this conversation. We want to make it as interactive as possible. So that was a very brief introduction to today. And without further ado, I would like to dive right into the topic for today. Michael, Todd, I would like to start by setting the very basics and perhaps asking a very general question. Can you explain to me and to our audience what the connection between proteins and disease is? Maybe Todd, you can start.
Sure. To understand what role proteins play in disease, you have to first know exactly the role they play in normal cellular biology and physiology. So proteins are the workhorses of life. Every cell in your body is packed with thousands of different proteins, and each one plays a different role. Some play purely structural or scaffolding roles, and those are akin to, let's say, beams or scaffolding in a building. Some function as molecular motors, for instance, during muscle contraction in your legs when you're walking or when your heart is beating. If you dig deep enough, there are protein motors driving each of those events. Some function to catalyze chemical reactions in your body, and we call those enzymes. And then finally, some are purely sensors or signaling moieties, and they monitor the local environment and they trigger changes so that the organism functions as a kind of collective. So genes are instructions for making proteins. Proteins are the end products, and proteins in turn create the structure and the molecular machinery that physically carry out the functions of cells and tissues and organs. So when a protein malfunctions, or some part of the way these complicated networks of proteins work together becomes unbalanced, the system doesn't operate correctly, and we call that disease. And most drugs target specific proteins, and they turn them either on or off in an attempt to rebalance that system.
Mm-hmm. So we have a protein in the system that for whatever reason needs to be turned on and off because it is related to disease. How does that specifically work? How does one tackle one of these proteins and turn it on or off? Michael, maybe you can take this one.
Yeah, Nuria, and hello from my side. I mean, yes, we can turn on and we can turn off proteins. Turning it off is actually, I would say, a long history, particularly of what we do in therapeutics, particularly with small molecules. Because if you think about a protein, something that is signaling or that is playing a central role in a living organism, it always works via interaction with other molecules. Also small molecules in living organism and cells. And there is a principle that was invented already in the 19th century that we call the lock and the key principle. So in other words, there is often something like what we call a binding site, a pocket or a lock in a protein, and there is a specific key to it, a molecule that binds very specifically to a protein, and that turns it on or it turns it off. And if we want to modify this function in a disease state, so when something went wrong kind of with the protein, we can use the same principle. We can try to find a molecule different from the one that is naturally occurring that will bind into that same lock, into that same binding site, into that same pocket, and will exert a function. For instance, taking the molecule, so to speak, out of business, inhibiting it, stopping it from working. And that's a key principle of small molecule therapeutics, binding into a pocket, into this lock, and modifying then the function of the protein that it is no longer, for instance, disease causing.
Mm-hmm. And here is where it gets kind of interesting, right? Because in theory, it's simple. We find this pocket or this lock in the protein. We design a small molecule that fits in there, and then we can stop any protein, so to say, from malfunctioning. But we know that this is not the case, and that not all, or that the theory is not so easy in all the cases. So maybe we can start talking about what makes some of these proteins undruggable or harder to treat. What makes some of these targets a little bit more complicated than others?
I can tackle that. I think it's important to understand, as Michael said, that proteins are three-dimensional objects. They have very, very specific shapes, and their surfaces have really different characteristics that range from smooth and nearly featureless to the presence of the deep grooves or pockets that Michael was talking about. Certain classes of proteins, particularly enzymes, tend to have a lot of those deep pockets, and therefore they've been easier to design custom fitting drugs that bind in them. So we call these druggable. However, many other proteins, about 2/3, in fact, of all the known proteins seem to lack those obvious deep pockets that characterize the enzyme class, and therefore we generally refer to these as undruggable proteins. And these so-called undruggable proteins can have roles in disease that's just as prominent as the enzyme class, but they've been more or less inaccessible. So I guess you could say they're special, that if you had a way to target them, you could tackle diseases in a new way. And that's what Vividion and Bayer are trying to do with our technology, is really target those smooth, nearly featureless protein surfaces without requiring those deep, structurally well-defined pockets. So we're using that technology to open up new possibilities for drugging new proteins, hopefully to treat diseases in new ways.
Mm-hmm. And would you say, so in a way, if I understand this correctly, we have these proteins that are very hard to access, and with this, or very hard to treat, and with this technology that Vividion and Bayer are advancing, we might be able to finally target those 2/3 of proteins that you mentioned we haven't been able to do in the past. And this would be also with small molecules, so with tools that we historically have used in the past?
Yeah.
Go ahead, Michael.
Yeah, thanks, Todd. Yeah, Nuria, that's exactly the point. And maybe to make it perhaps a bit more transparent, or to give you an example without going into a technical detail, I vividly remember my very first project in my time at industry. When we had a protein, we were very convinced it played a central role in a disease. It was heart disease. But the pocket of this protein was shallow, it was very smooth, it was like a bathtub, I would say. Of course, and a bathtub is, of course, I mean, we are talking about the microscopic world here, but it was far too big simply that a small molecule would fill it, and there was nothing where the small molecule could hold itself, where it could really bind to and make a tight connection. And that's now the difference with the technology that Vividion has developed, that we find that we are able to tackle proteins not needing these big and shallow pockets, but finding new ones. Mm-hmm. And think about this. As I said, we were very convinced about this protein, that if we were to find something, it would have a high likelihood that at the end we could help patients with this. But we simply didn't have the means to do it.
Maybe let's talk about this, because you mentioned now this was at the beginning of your career, which might have been a bit ago. So in these years, what has changed? What makes the time now particularly exciting for, for this type of targets that are so elusive? What is special from today?
I think we don't have so much time, right, Nuria? But there's a lot.
Maybe we can try to summarize it. So.
There's big advances in chemistry and biology, and that all converges now. There is big advances in data science and computing power that helps us all in this. I mean, just reflect. I was thinking this morning. The 2 Nobel Prizes in chemistry of the last 2 years. One went to chemists who shaped a methodology that helps us now coming up with new molecules much faster in a better way. And before that year, it was the Nobel Prize for the CRISPR technology that helps us, apart from what you can do perhaps in the field of cell and gene therapy with it, but it helps us understanding biology much, much better. And that's also the root causes for diseases.
It sounds definitely very exciting. I would like to hear a little bit more from Todd, how this convergence of biology, disease understanding, chemistry, computing power, how has this translated into the technology that you are advancing, or that the team at Vividion is advancing to unlock this undruggable target space? How specifically does this work?
Yeah, I think our platform really is powered by 2 fundamental technologies that combine to make it possible to target these previously undruggable surfaces. So the first technology is a specialized type of chemistry that Michael talked about, and that allows us to exploit a very different surface feature on proteins instead of deep pockets. So our compounds use the presence of a certain amino acid building block called cysteine that's a regular component of proteins. In fact, these cysteine surface features are far more abundant on classically undruggable proteins than these deep pockets are. So we have the ability to extend our reach into that 75% portion of the cellular protein network that was previously just not possible. And that alone is a game changer for the kinds of proteins we can target, but employing that approach in a way that's practical and efficient and scalable and gets you to compounds with drug-like properties requires a second technology. And that second technology is broadly called chemoproteomics. And that's a very broad array of techniques that utilizes very specialized instruments in technology to detect small molecule interactions in proteins. In our case, we're looking for those cysteine feature-dependent binding events. And it's a special technology because it has an extremely high level of accuracy and precision, so we can reliably detect those exactly which proteins or compounds are interacting with. The resolution is high. We can even tell exactly where on the target proteins the molecules are engaging. This technology has an amazing ability to filter through complex mixtures of proteins to focus on only the important binding events. So it can be used to assess compounds directly in cells that are packed with thousands of these different proteins all at once. And finally, the methods allow for massive parallel processing and detection. So that's where the computing and technology hardware improvements come in. There are so many thousands of potential interactions that can be examined in cells with just within a few minutes for each compound that we want to investigate. So put it all together with the novel chemistry and the ability to go to these new areas and the technology to actually examine that with a high level of detail. And it's a lot like searching for a very rare object with a new way to do that, with a new level of efficiency.
So in a way, I will try to put this in a 30-second summary, very basic, and you need to tell me if I got this right. So
Okay.
basically, if I understood correctly, there's a significant difference from Vividion's technology, which is we're not looking anymore for these deep pockets, which sometimes in hard to drug targets are not there. We're looking for another feature, the cysteine residue, that seems to be rather common. So we're changing what we're looking for here. And you have found a way to basically be able to scan many possible candidates that could interact with these and find the needle in the stack of hay that actually does the job by applying advanced computing power, detecting methods in a very, very, very quick nutshell, so to say.
Yeah, that's a very good summary. It's the
Okay,
blending.
well, thank you.
Yeah.
Okay. One thing that we have kind of talked about, but that I would also like to go into more detail is commonalities of these proteins that are hard to drug or elusive. Are they more common in one disease than in another? Is there a specific area where these seem to be a little bit more tricky, or is it a general thing? Do we see them spread across diseases?
If I were to answer this, you can find them as root causes for many diseases. It's not that they are, that we find them exclusively in the one or the other areas. I mean, I would know about examples from the field of certain cancer types or of certain inflammatory diseases or also diseases like heart diseases or kidney in kidney malfunction. So in principle, as we are talking about basics here in biochemistry, in the way how proteins interact, how proteins have pockets and function, basically you can find that in proteins causing many different diseases.
So I will try it again and try to summarize this. So basically, this technology that we're discussing is not limited to one specific disease. It would really allow us to look at a broad range of diseases, wherever that protein is, so to say. We just need to make our pick.
Yeah,
and this is simplifying.
Yeah, and I would add that since just about every aspect of a cell function involves proteins, essentially every disease, at least in theory at some level, involves these proteins. And since we're expanding the number of proteins we can interrogate to that 2/3 of that were previously inaccessible, it makes a lot of sense that if we want to unlock new opportunities, there would be more of them in each disease area that we might be able to go into. So just the technology alone just gives us many more choices. In some disease areas, there are not a lot of choices. And so getting that new territory to search is just critically important.
Todd, do you think you can give us a specific example? Is there something specific that you're thinking about where maybe there are not so many options, where it would be helpful to look for other options?
I can give you an example. Let's say even on a druggable target, sometimes inhibiting a single target in a deep pocket inhibits its function everywhere. And sometimes that's not desirable. It comes with side effects. It could be that what you'd really like to do is just inhibit a specific aspect of its function that is driven by a specific location, say far away from the active site. And if you could do that surgically, because you had this technology that allowed you to interrogate specific surfaces, you might be able to selectively just turn off one of several nodes that protein is important in. And so I think, that gives you therapeutic optionality. Maybe in some diseases, the toxicities that come along with inhibiting the entire protein really present a serious barrier in being able to surgically do that, just simply, again, because you have more options mechanistically and where on the protein you can target them. With our technology, you're able to expand the universe in new areas.
I like that, to expand the universe. Maybe let's talk about expanding the universe, because we have talked a lot about switching off proteins or switching them on, so inhibiting or activating proteins. But over this week, precisely on these articles that I was talking about on our LinkedIn channel at Bayer, we have talked a lot about what we call new modalities, so different ways of tackling these undruggable proteins. And we have talked about PROTACs, we have talked about RNA targeting small molecules. And I would like to hear your thoughts a little bit about how all these different tools are coming together, and what also are the possibilities, or what possibilities do these tools bring to us in addition to what we have already been discussing?
Yeah, in this picture of, to stay in this picture of the universe, Nuria, that you mentioned, I mean, there is a huge opportunity for us. I mean, if we tend to stay with the universe, or let's say with our solar system picture, and say, I mean, the moon is what we at current address, but there is the Earth, and there's particularly the Jupiter is the biggest planet, and I mean, the Jupiter is approximately the size of all the proteins, and also in the RNA world, that we could address. So there is a huge opportunity for us to address processes, to address proteins, be it really on a protein level or on RNA level, that cause disease. And now the question is how to do that. So we heard from Todd the approach from Vividion on chemoproteomics, and with all the data science also that is behind it, helps already to address part of it. And it's particularly targets that we feel we have a very strong connection to a disease, and that makes it so super exciting, I have to say. Yeah, now all of a sudden that you can address certain targets, but there might be others, and chemoproteomics, it might help there or might not help. And the other modalities that we talked about this week in the various channels, they might be in a way have different properties, and might be helping to use those. I mean, think about RNA targeting. That's a very, I would say, intriguing, very exciting technology that already on an RNA level, you kind of interfere with this world of disease-causing proteins. Think about this. It's a totally different world. It's a totally different way of addressing, of binding to biomolecules that play a role. You know, as a chemist, you have to think in a totally different way. You are used to interact with what Todd already mentioned, amino acid structures, and now all of a sudden we talk about RNA structures, so nuclear bases and so on. It's totally different. It opens a totally new world. So what I see, and it's basically also matching, I would say, my experience, that there is never this one and only technology, modality, however you call it, but it's always the sum of the different technologies that you have. I'd say this all together will help us to address that universe of disease causing, let's call it molecules for a second. Yeah.
So we can target proteins, we can target RNA, which is the step before proteins. What else can we do? What else is there left to do? Todd, maybe you can give us your thoughts. What would be the next exciting thing?
Well, you know, and I can tell you that the Vividion chemoproteomics platform, I guess you could describe the covalent molecules as their own modality, although, you know, those have been around for a while, so that part's not new. But you can combine our ability to find these unique compounds and go after unique proteins with some of these modalities, like the degrader modality. You know, I think most in the field, maybe without diving too much into that technology, but it essentially independently binds to a target, and it independently binds to E3 ubiquitin ligase, which is a kind of enzyme. And if you induce the proximity of those two proteins, you can get that ubiquitin ligase to tag the target protein in a way that directs it to the cell's garbage disposal system. So it's really a pharmacological method to induce proximity. But from a practical standpoint, and I think the field understands this, that there's a very real bottleneck limiting the true potential of the degrader modality, because you still need to be able to discover those new target binding molecules. And if you're limited to only deep pocket binders, that's the only, that's likely your limitation of the kind of molecules. And so again, here Vividion and Bayer are in a unique position, use the platform to find these unique target binders that our molecules can bind to, these proteins that may not be inhibitory on their own, and we have the flexibility to convert them to degraders. So we can blend those two technologies together when it's to our advantage. And maybe not to be too long-winded here, but an exciting example to think about is that some E3 ligases are expressed in only some tissues. And that means if you require the E3 ligase for your bioactivity, it has to be present. And sometimes you want to avoid certain tissues to avoid off-target toxicities. And that means if you have novel ligases that you can pull on, you can maybe make small molecule drugs that operate in tissue-specific manners.
I'm getting excited about all these opportunities with finding new small molecules, finding potentially new degraders as well, with chemoproteomics. And we got a question from Mona Talari, who actually would like to learn a little bit more about the detection techniques. So we talked about, Todd, you gave us a little bit of details about how you could scan all these molecules, again, a specific target or a set of targets. How does, maybe very generally, how does the detection work?
Okay, let me try to keep this non-technical. The technology is actually, and how we find these things is not all that complicated. The instruments are, but how we do it isn't. So imagine a process where we have our compounds arrayed out in a plate, one compound per well. We incubate that compound with lysates or cells. We give it time to interact with everything that it diffuses and collides with. And in some cases, if there's solvent-exposed cysteines and those are positioned just right next to a shallow surface or a pocket, our compound can react with those. And then in the second step, after we've allowed the compound to do that, we add what's called a cysteine reactive probe. And that's really something that is so electronically hot or chemically reactive, it just, whenever it collides with a solvent-exposed free cysteine, it reacts with it. And our probes have a linker and what's called a biotin on them. And that coats the proteins everywhere where the compound is not bound. We chew those proteins up. We can use that biotin tag to pull out the fragments of the protein where it's reacted. We wash away those that are not relevant, and we shoot those down a mass spec and detect each unique peptide. And imagine 4,000 different signals that you can reliably view. And anytime your compound reacts with one of those cysteines before your probe got to it, that signal disappears. And so you just look for dropouts in the screening method, and that tells you where your compound is binding on the protein. And with the right informatics, you can do that in very high throughput and figure out the protein that your compound is uniquely reacting with, and even the cysteine and the site on that protein where it's reacting.
So maybe as a key take home for those that are not so deep into the technical details, you can scan or specifically locate not only to what protein this residue has made a bond with, but also where specifically within that protein.
Yes, that's correct.
Wow.
We specialized instruments to detect those events.
Maybe I would like to take the conversation to, we've talked a lot about how we find these pockets, how we might find candidates there, but I would like to talk a little bit about what happens after. So once we have, say we have this perfect candidate that we have, how different is or will be to bring these candidates to a patient down the road. So in comparison to what we would call a traditional treatment or a traditional pill, so to say, how different is this going to be? How can we expect that this will be different?
Well, maybe I take it, and I think Todd already mentioned it. The molecules that Vividion is working with are not a totally new modality. This type of covalent binders are known since many years. The way Vividion identifies them and also uses them to identify novel pockets, that's new and that's super innovative. But in principle, the modality is known. It's a small molecule. There are some gradual differences to, let's say, non-covalent small molecules, but to the scientists that work in the industry, that work in medicinal chemistry, that do the pharmacology, it's not so different. Yeah. So at the end, what we aim for is a molecule that you can give via the oral route. So as you said, Nuria, as a pill, and that you also develop in clinical tests, in clinical trials, in a very similar way, like usual small molecules.
Mm-hmm. So in a way, it's getting better at something that we kind of know what to do, or finding new ways of doing something that we kind of know what to do. Yeah.
And that's another powerful component if you think about it. I mean, there are other modalities. I think Todd mentioned, for instance, the protein degraders, PROTACs. It's sometimes a bit more tricky. Yeah? They're bigger, the molecules, not so easy to find. You have to do a lot of optimization to get them available via the oral route. So there, the modality is also new. Yeah, which of course, I mean, we are also excited about PROTACs, but it poses another risk. Yeah, for the technical probability of success. And here with the molecules that Vividion finds, I'd say this risk is rather very comparable to what we do with other small molecules.
Yeah, I think these drugs will seem a lot more familiar to patients than not from their perspective. This is kind of like buying a new car. You may not understand or be privy to all the details of how that engine is working, but all cars are pretty similar, right? You get in them, you hit the gas, you move the steering wheel to drive it.
Thank you for that visualization. I think that really helps to understand what the differences might or might not be. One thing that we have been talking a lot, actually that has been a theme, an underlying theme throughout the conversation, is that we do have people from Vividion and from Bayer in this conversation. And one of our listeners, one of the people in the audience, Amelia Elizabeth Rose, is asking how important collaboration across the pharmaceutical world is in order to truly unlock this potential for drug development. So clearly, we have a very specific example of collaboration here, and I would really like to hear your thoughts about this, maybe from both of your perspectives, actually.
Well, I think one way is just continue to lead. I have to say, Vividion in particular benefited from, the kind of covalent molecules that came before, the EGFR receptor inhibitors, the BTK inhibitors, the KRAS G12C inhibitors, and that kind of that told the field that we could very successfully and safely in general use this covalent modality. And so, and now we're bringing slightly refined versions of molecules like that are hitting different proteins, different classes of proteins, maybe some classes that haven't been heavily drugged before. And so just creating the template for others to see, I think, is a really important role. And we're just completely focused on moving what we know is working now forward as fast as possible, expanding that into different disease areas. We will and be committed to publishing those results at some point.
So definitely there's a component of building on research and contributions from people who have come before. Michael, maybe you can talk about this collaboration specifically. How do Bayer and Vividion make the most out of this collaboration? What is it special? Why did you guys team up together?
I think what we saw, I mean, let's say a very good and fruitful collaboration. I mean, there's a lot of ingredients to that: trust, the willingness to collaborate, excitement about what the other's doing, and I think that is a bit also the nature of, working as a scientist. Every day you read novel publications, you see cool stuff that others are doing, and you start thinking like, what if we were to team up? And there's always an equation that looks unlogical from a mathematical point of view, but that is 1 + 1 and 1 equals more than 2, equals 3 or so, because you find things that are just getting better by teaming up. There's so many examples that I could allude to here in the collaboration between Bayer and Vividion. I mean, from a Bayer side, I'm also always super impressed by the technology of Vividion, by the speed, the precision of how the colleagues at Vividion find the molecules. But when it then comes, let's say, to the next steps, to getting ideas how to develop it in the clinics, also perhaps with some aspects of optimization of the chemical structures, of some details in pharmacokinetics and the like, we can use the Bayer know-how to help and to support here and to make the drugs even better and to bring them also finally to patients. I mean, there's also big excitement, I know, at the Vividion side, bringing finally the compounds to patients, and that is supposed to happen very soon. And I think here we have big chances to collaborate and to speed up things, also to help patients faster than we could do it, both of us working alone. Yeah. In general, I mean, I think the pharmaceutical world, and I thought that this question is broader than just the collaboration between Bayer and Vividion. I think we all benefit very much from collaboration. There's so much knowledge being generated at the moment all over the world. It's incredible. It has never been in this speed and in this depth and quality that you always think, who could be a collaboration partner to make things working, to bringing novel medicines to the patients. And we at Bayer, we've worked with many institutions, academia, biotech, pharmaceutical companies to make it happen. Yeah.
Michael, I think, from my perspective, I think I could give a kind of a granular response: screen more compounds faster. And one of the limiting bottlenecks for us is having more compounds that give us more options to screen. We've been working on that technology to screen faster and in highly multiplexed manners, but more compounds. And so, when we fused with Bayer, we automatically were granted access and had that capability to take some of their existing covalent library, which in many ways is similar to ours, and push that into our chemoproteomic screening methodology. So we just increased the size of the starting substrate for our searches, and that alone, I think, is really important.
So really increasing the space of that universe that you have to tap into, so to say, hopefully pushing the frontiers of what can be done. I hear a lot of excitement coming from both of you, and unfortunately the time of the conversation is coming to an end. But before we wrap up, I would like to ask both of you, Michael and Todd, if you would have to summarize very briefly, what makes you personally excited about this particular time for drug discovery, and why should we be excited about it? So, Michael, maybe you go first.
Yeah, I mean, I mentioned already, it's the conversions of so many novel science and novel methodology. And now, I mean, thinking about what we talked today, drugging the undruggable, you almost suffer as a scientist over many years from knowing there are good targets or good proteins that you could address and have a big role in helping patients with certain diseases. And it was a bit frustrating over time to not being able to do this, despite of all the progress. And now all of a sudden we live in these times where we have all these new methodology at hand. And that makes it so exciting. All of a sudden you are in a position where you can not just dream, but really see a path forward for all those undruggable targets and all of those diseases that are so heavy to treat. And that makes it super exciting.
Yeah, I think that we've really entered a new era. It feels like a renaissance in terms of small molecule drug discovery. It's the right time. The technologies have matured now, and we have real actionable opportunities that we can advance. And it just feels like something tangible is different today than it was 5 years ago. The convergence of all these technologies and opportunities are putting us in a new era, I think.
That's a great note to leave it. Michael, Todd, thank you so much for having joined us today for this conversation. To our audience, thank you very much for following up today's LinkedIn Live. I want to encourage you, if you haven't had the chance yet, to go to our profile. If you want to learn more about PROTACs, about chemoproteomics, we will put the links in the chat to the blog articles that we were talking about at the beginning. And if you're interested to learn more about these exciting themes, what opportunities might be there for you, please do check Vividion's buyer's website and keep your eyes open. And with that, I would like to thank you very much for joining us today and wish you a lovely rest of your day, wherever you are. Thank you very much.
Thank you.